So, hello and welcome to week 15, our final week in ESTEP of new content in this course. So with that, let's dive into the slides. We're going to be investigating fossils this week. So I'm going to keep it pretty straightforward. We've got three main questions or topics here. What are fossils? How do fossils get preserved? And fossils in Minnesota. On the right, you'll see a time scale that is the sort of the kid-friendly pictorial rendition. And as you remember from our investigation of geologic time, we have the oldest down at the bottom and the youngest up at the top. What I want you to pay particular attention to here is if you look at the area on the left, you'll see pictorial renditions of fossils. We only really start seeing any hard-bodied fossils near the base of the Paleozoic. Remember that the Paleozoic, Mesozoic, and Cenozoic make up the time chunk that get grouped as the Phanerozoic. And it's really only in the Phanerozoic that we see hard-shelled fossils. Prior to that, there were soft multicellular organisms later in the Proterozoic, but no hard-bodied fossils. This is called the Cambrian explosion. Okay, so let's dive in. So, first of all, what exactly are fossils? And we can divide fossils into two main groups. First of all, we have body fossils, and these are, as the name suggests, the remains of body parts of ancient animals, plants, and other life forms. So on the left, you can see the bones from an Archaeopteryx, and on the right is a sample that could come from the Decorah Shale in Minnesota, exposed in southeastern Minnesota and in areas around the Twin Cities. So these display good examples of body fossils. In the case here, we're looking at the shells that the organisms built to protect themselves. Or here, this Cheerio-looking ring that is basically the stem of an organism. Now, the other type of fossil in our nice, tidy organization here is trace fossils. These also get called ichnofossils. Now, what are trace fossils? These fossils, or they're more like patterns in the sediment that provide evidence for movement or some kind of other activity of an ancient organism. So these actually get divided into four or three different types, and then, of course, subdivided even more. Movement, predation, and digestive fossils. So movement, I haven't got examples here, but that would be footprints like dinosaur trackways. If there's evidence for one organism preying on another, that would be predation. Digestive would be more, or feeding would be more like the patterns that you see here. These are planolites, and we actually see a lot of these in Minnesota, in the sedimentary rocks. These are effectively trails left by organisms that were scavenging the seafloor sediment for nutrients. They ingest everything, and then they effectively poop it out or scoop it away behind them, leaving these tracks. So this is something you are most likely to encounter or may have seen. You'll see this or some evidence of these trace fossils in some of the facing stone at Target Field, if you go there. Okay, so just a bit of an overview now. Remember, down here is old, up here is young. Prior to the Phanerozoic, in other words, prior to 541 million years ago, no hard-bodied organisms. Then, as I've mentioned before, and I'm mentioning again, came the Cambrian explosion. And this represents the biodiversification of multicellular organisms and the time at which organisms started to develop shells. And what I'm not going to dive into, but the fossil record provides really important evidence for evolution because the record provided by fossils shows how life on Earth, well, first of all, was once different to life found on Earth today, but it also provides a detailed record of how organisms have evolved and changed over time. And some have become extinct, and then we see the addition and or development of new organisms. So some of the really key steps during the Precambrian, in other words, the Archean and the Proterozoic. So there were early microbes around 3.5 billion years ago. So that's way back in the Archean. But the biggie is the evolution of cyanobacteria. These are the first photosynthesizing organisms. And they, because they photosynthesize, produce oxygen. So this is called the Great Oxidation Event. It introduced oxygen into the atmosphere and into the water. And actually, that's one of the reasons that some of those older microbes became very diminished or died out because they could not tolerate oxygen. So as we're moving forward in the Proterozoic, before we get to the Cambrian, there is evidence around 800 million years of multicellular life having evolved. And this involved some sponge-like organisms that are preserved in various ways in the fossil record. And then as we move up towards the Cambrian, there's a quite complex suite or group of soft-bodied organisms that get called the Ediacaran or Ediacaran, depending on which side of the Atlantic you grew up. And they are soft-bodied, so they still don't have a shell, but the records in the sedimentary rocks indicate that they are definitely a lot more complex. Now, at that Precambrian to Cambrian boundary, those Ediacaran organisms became extinct. And that's when we see this Cambrian explosion. So the sedimentary rocks in Minnesota essentially span Cambrian, Ordovician, Silurian. As you head south towards Iowa or into Iowa, you get into Devonian. So there's actually some very nice Silurian Devonian road cuts in Iowa if you want to go and explore them. So the simple overview of a geologic timescale divides the Phanerozoic into our three time chunks: Paleozoic, Mesozoic, and Cenozoic. So Paleozoic, that's derived from Greek and effectively means ancient or old life. And the most common fossils found in Paleozoic rocks in Minnesota are your brachiopods. There's also a whole bunch of other ones. We'll look at that towards the end. After the Paleozoic, we move into the Mesozoic. That's from the Greek for middle life. And then we move up into the Cenozoic, which is from new life. And most of the organisms that we see today, the mammals, for example, all evolved during the Cenozoic. I've put a whole bunch of resources into D2L that go into detail about the different fossil types, etc., etc. So as you've noticed on our column here, not only do fossil organisms evolve over time, but Earth has also experienced some really, I'll say, life-changing extinction events. So essentially what happens is there is a widespread and rapid decrease in biodiversity. So something happens to kill off a whole bunch of organisms. And those extinctions, well, there's 5 main extinctions that are recognized, but the really big ones that tie in rather obviously with the stratigraphic record are the extinction events between the Permian and Triassic at about 252 million years, and then the extinction between the Cretaceous, symbolized K, and Tertiary. This is where I'm just going to remind you that the Cenozoic is actually divided into the Tertiary, which includes the Paleocene, Eocene, Oligocene, Miocene, and Pliocene, and then there's the Quaternary, which is made up of the Pleistocene and the Holocene. And as you'll remember from our glaciation module, we now know that the Quaternary goes to about 2.5, 2.6 million years ago. Anyway, the big deal here is we've got extinctions at the Permian-Triassic boundary and the Cretaceous Tertiary. The Cretaceous Tertiary extinction event is the one that's associated with the demise of the dinosaurs. That's the one that you and most of your students probably know most about. And there's a lot of discussion about the causes for these extinctions. And yes, meteorites or meteors are involved in some of those causes. Now, next, let's think about how fossils get preserved. We can divide this into 2 types of, or 2 groupings, unaltered remains and altered remains. So essentially, if we see unaltered remains, what we're saying is that essentially the original composition and structure is preserved. So for the most part, this is something we see in fossils from the Cenozoic. And what I picked up here is by freezing, mummification, or preservation in amber or tar. So it's not uncommon for gold miners in Alaska to find frozen woolly mammoths. This is a woolly mammoth calf that was frozen in permafrost that some alluvial gold miners found. Mummification would just be the drying up of material. And a wonderful example of that would be Otzi the Iceman. I decided not to include a picture of that. Then there's preservation in amber or tar. So on the lower right here, we've got a bit of tree resin, but what I want, and that is called amber. And inside that tree resin, we have some insects that have been preserved. And they've been able to extract DNA from some insects that have been preserved inside amber. Another example of unaltered remains is when organisms get trapped in tar, or you can just think petroleum material. One of the most wonderful examples is the La Brea tar pits in LA. So in the middle of LA, there is this area called the La Brea tar pits. I haven't gone there, it's on my list, but essentially organisms fall into a area where there's some kind of oil seep, and that allows for preservation of them. So we've now looked at unaltered remains. Now we're going to think about altered remains. And when we say altered remains, what we're saying is, okay, there's been some kind of change in composition and or structure of the original materials. And we've got 4 types of alteration. Now, the first one is permineralization. So in this case, I want you to look at what you may identify as petrified wood. That is the common term for technically what we call permineralization. And in permineralization, fluids fill in empty spaces in whatever the original material is. So your petrified wood is typically replaced by silica, SiO2, that has been moving through the system. Often that's produced as a result of heating somewhere that has heated some fluid that is silica rich. So that's how you get your fossilized forests or your petrified wood, technically permineralization. Now, there's also recrystallization. So recrystallization, if you look at the fossil on the lower right here, you can see in this pinky area, the part that was originally the shell has been, in this case, replaced by probably calcite and or some other mixtures of minerals. You can see that the empty spaces in this organism were filled up with sediment. But when the shell material gets replaced by some other mineral, sorry, not replaced, wrong word, when it recrystallizes. So we have a shell that was carbonate originally, that carbonate recrystallizes. Now, we're going to go up to the top here, and this is replacement. In this case, our shell material has been replaced by pyrite, iron sulfide. So for some reason, where the shell is on the seafloor in the sediment, it's in the right kind of reducing conditions to allow pyrite to replace the original shell material. One also sees pyrite replacing sand dollars. That's something you commonly see in rock and mineral shops. Finally, carbonization. So if we have plants that have been deposited, here we're looking at a bunch of fern fronds. What happens is a lot of the organic material will actually degas, and it just leaves carbon as the remainder of that organic material. And that's carbonization. So we've looked at various ways that fossils get preserved. There's one other piece we need to think about now. So imagine you've got some kind of shell, and here we are with what it will be. This is some kind of bivalve. And what are you looking at when you look at a fossil? So there's two important terms, molds and casts. So the simplest way of thinking about this is if we treat the original shell or other material as having been dissolved or has decayed. Essentially, it is no longer. Now, what do we end up looking at? First, there are, I'm actually going to do this the other way around. First, we're going to consider casts. And a cast forms when the original impression or inside of the organism is filled with sediment. So the easiest way to think about this is, okay, here we have our original shell, and that shell gets buried and is likely, the shell material is dissolved, but the outline of it gets preserved in the sediment. And what happens is all the sediment that was inside that or formed part of the shell preserves the shape of the shell, and that gives us a cast. So the cavity is filled to form a cast. So that's our casts. Then we get molds. And so in the case of a mold, we have our organism, animal or plant, that's buried in the sediment and decays. And it leaves behind the impression of the organism. So there's various ways this can be diagrammed, and this is a potentially confusing diagram. But so essentially just consider we have a shell that is buried. It may recrystallize with sediment inside it and the sediment around the outside. Now, what then happens is if we end up looking at the sediment that collected outside that shell, we're looking at an external mold. It's the shape of the outside of the fossil. On the other hand, if we end up looking at the shape of the inside of the fossil that is preserved in the sediment, that is an internal mold. And as I hope you've learned to appreciate over the past 15 weeks, nothing is tidy and simple, and there are lots of gray areas. So we often see a combination of some of these features when we're looking at fossils. Okay, so let's just practice this. I want you to look at these two images and think about what they might be in terms of casts versus molds. Or also just what has happened. So over on the right, when I look at this, can we see the actual shell? No, not at all. That is completely dissolved. So what we're looking at here is an internal mold. And in this case, this is an internal mold for a gastropod. That's the word for a snail of some kind. Now we're moving over here to some trilobites. And I put the trilobites in to remind me to say that the trilobite skeletons are made of chitin, which is a dark gray black material. So if you're looking for trilobites, particularly in some of the Minnesota rocks that you may investigate, you will usually see dark gray black fragments. It is rare to find any full trilobites. So, knowing what I've just said about the trilobite skeletons, when you look at this sample, what I want you to recognize is that you are not looking at the actual shell material. What you're looking at is the impression of the shell. So effectively, this is some kind of internal and or external mold in this sediment. So I just mentioned trilobite shells. What you can see here is a partially replaced trilobite. The dark material is the chitin. Now, I also want to just mention here that if you are buying or your school owns any teaching samples of fossils, what you most likely have are synthetic casts made from plaster of Paris. So essentially what they do is they take the original fossil, they put it in something to make the shape, and then they put plaster of Paris into that shape to make a cast. So that's why samples that you may come across or you may have been taught with don't look anything like the real thing. They're just plaster of Paris casts. Okay, so finally, there is a really good Minnesota at a glance leaflet on fossils, fossil collecting in Minnesota. And the focus is on the sequence in the Twin Cities. Sorry, outstate folks. There are many localities in southeastern Minnesota where you can see some of these same units. So I put the link to the Minnesota at a glance or a PDF of it into D2L for you. What I've also done is put a reference up here to a blog. And the reason I picked this blog is there's actually lots of images of fossils. Justin Tweet has done an amazing job collecting those images, so it's a great place for you to look for Minnesota in particular fossils, and it's easy to read. So getting back to fossils in the metro area, the unit that you are most likely to find fossils in is the Decorah Shale. Now, it's a really interesting unit because it's called the Decorah Shale, but it's made up of shale, and there are limestone or limey lenses in it. And for the most part, those limey lenses are what's called a coquina, or I'll just call it a fossil breccia. So what it's telling you is that there were all these critters living probably somewhere maybe further up the slope in the ocean, somewhere closer to land. then there was a big storm. They all got broken up because the fossils here are mostly broken up. There are some whole fossils, but for the most part, they're kind of broken up. And they were deposited as these layers of broken up fossil material. And the image on the left here is a summary of the Ordovician fossils found in Minnesota. So the Decorah is Ordovician in age. And you will be likely to see brachiopods. You are very unlikely to find a whole trilobite. What you are most likely to find is just a trilobite spine or a bit of a tail. Finding an enrolled trilobite would be a major coup. So, whoops, okay. So those trace fossils, as I've mentioned, crinoids, they are the organisms that look like plants but aren't plants and have stems, and those little Cheerio looking things are part of their stems. There's gastropods, think snails. There's corals. There's a little horn coral here. Cephalopods, these are squid-like organisms. And then there's pelecypods. So these are also called bivalves. And no, wait a minute, pelecypods. And then we've got bryozoans. The bryozoans are colonial organisms, and each of these little divots that you see are on the surface of this sort of tree-like structure, and an individual organism lives in each of the little divots that you see here. So the image here is the best one for comparing to what you are likely to see. There are some bryozoans that get called gumdrop bryozoans, not the technical term. They look kind of like little gumdrops. So that's something you could see if you collect some fossils. So with that, I'm going to leave you to investigate fossils in more detail. But thank you very much, and we'll be in touch. It's been a joy working with you. Thank you.